Okay, so in this podcast, we're going to be talking about lymphocyte receptor gene rearrangement. So in class, we may or may not have time to discuss the evidence and discovery of VGJ recombination, but for now, what I'd like to do is walk through the arrangement of gene segments and the process of recombination. Okay, so we're going to start with the organization of the gene segments. So let's get right into it. So organization. And we've talked about in class how there is a heavy and a light chain. So light chain and heavy chain. Okay, and the light chain comes in two flavors. So let me just, got a lot of color coding going on in this podcast. So we have the kappa chain, or kappa, and we have the light chain, or sorry, the lambda chain. And each person has two of these chains, so this allows for 4 possibilities to make a productive rearrangement. And we'll revisit this concept later when we talk about the B cell and T cell receptors. There are these gene segments present on the light chain and the heavy chain, and the gene segments are called the V, D, and J regions. Okay, so I'll tell you a little bit about their organization on these chains. Okay, so on the kappa chain, we have these boxes that represent a single V segment. Okay, and this number goes on because for the kappa chain, V kappa, there's approximately 41 of these gene segments. And this is an estimate. And then the next part are these J segments. And there are 5 J segments on the kappa chain. And then we have 1 constant region on this kappa chain. Okay, so something to notice right away is that I said there's V, D, and J regions, but the light chain only has V and J. Okay, now the lambda chain is arranged a little bit differently. So you still have these various V regions, so V lambda, where the V on lambda, there's about 33 of these gene segments. Okay, so that's pretty much the same. Let me make sure this is all on camera. And then this is just a little bit different because they do have the joining and constant regions paired up in this case. And you won't have to know exactly how this is arranged, but it'll help you kind of understand the importance of having these gene segments. Okay, so there's around 33, as I said, V regions, and then the J regions, there's 5. Okay, so some key concepts when it comes to the light chain is that there's 4 chances, and I'm going to write this down here. So light chain, key concepts. Okay. There's 4 possible opportunities for productive rearrangement. There's V and J segments only. And we can start to talk about how these segments can influence antibody diversity or T cell diversity. Okay, so you can multiply the number of gene segments to get the possible number of combinations. Okay, so for the kappa chain, you can multiply 41 by 5, and there are 205 combinations that you could, possible combinations you could get from just combining the gene segments. And we'll talk about lots of ways, other ways to introduce diversity into the lymphocyte receptors. And then for the lambda chain, it's 33 V segments times 5 J segments for 165 combinations, okay? So gene segments equal diversity. Okay, so those are the key concepts there. So I'll highlight that. And let's talk about the organization of gene segments on the heavy chain. Okay, so heavy chain, we have V regions, V heavy, and this goes on, and there's around 45. Okay, and then there's D regions, or D gene segments, so D heavy, and there's 23 of these, and these are estimates. It can vary by individual person. And then there are J gene segments. 1, 2, 3, 4, 5, 6. J heavy, N equals 6. Okay, so these ones keep going, and these ones keep going. And then there's a few different constant regions that will dictate what the isotype becomes. So let me just make this a little bit longer. So there's constant region for mu. This was the gene segments for a B cells is kind of what we're using as our example. And then there's the delta that makes IgD. And then there's a few versions of gamma constant chains. So this is C gamma. This is 3, 1, 2A, and 2B. And this is constant epsilon and constant alpha. So you can calculate, right? I mean, there's 2 copies of these. So there's 2 chances to make a productive rearrangement. So this is our heavy chain key concepts. Right, there's 2 chances. There's V, D, and J segments, and the diversity can be calculated by multiplying the V, D, and J all together. So 45 times 23 times 6 equals 6210. Possible combinations for the heavy chain. Okay, so a lot of combination, or a lot of diversity by just pairing together different gene segments, and so we're going to pick one of these, and one of these. But also there's diversity introduced by pairing together the heavy and light chain. So if this is our light chain with our V, J region, and then the constant region here, right? And then we have a heavy chain here with V, D, and J up here, and then the constant region right there. We introduce additional diversity by just the pairing of these two different proteins together, or peptide chains, okay? So heavy and light chain pairing also increase diversity. Okay, so that's organization. And the next thing we're going to do is talk just a little bit more about how this process is regulated, and then we'll go step by step into the mechanism. What we're going to do eventually is recombine the V, D, and J gene segments into one piece of genetic information, okay? And as you would predict, if you did this in any cell besides this particular cell, the consequences might be disastrous. So this is a highly regulated and sequential process. So let's see, highly regulated and sequential. So the highly regulated part we will talk about more in the context of T and B cells, but certain proteins are turned on at very specific times. The RAG enzymes and the TDT enzymes, they're only expressed in certain cell types. And also this involves chromatin remodeling, so it's very regulated, and it's also sequential. And so in a B cell, for example, we have our heavy chain being made first, and so here's our an example of our heavy chain. Right, the VDJ is actually going to be right here, and this is going to be, the rest of this is going to be our constant region. And what first happens is that this goes to the surface paired with two proteins, one called V pre-B and one called lambda 5. And together this makes the pre-B cell receptor. And the purpose of this pre-B cell receptor is to send this heavy chain to the surface of the cell to make sure that a productive rearrangement occurred in the heavy chain before you spend energy making the light chain. Okay, so and then of course the light chain can be rearranged once you've made a productive heavy chain. Okay, so that's an example of how it's sequential. Okay. So how do we find the DNA that we want to recombine, okay? And this is largely guided by these DNA motifs called recombination signal sequences, okay, or RSSs. So this is recombination signal sequences, or RSSs. Okay, which is commonly how I'll refer to it as. Okay, so here's showing you an example of the heavy chain. Okay, so we have our heavy chain here, and we've got, I'm just going to put one on here. So we've got a V gene segment, a D gene segment, and a J gene segment. Oh, should have been blue. Okay, so flanking these segments are RSSs, recombination signal sequences, okay? So I'm going to represent those by little triangles. Let's pick a color. Just put them here. Okay, so these will represent my RSS signals. Oops. So these signals are very uniform. Okay, so if we zoomed in and we looked at the sequence, here's what would be in a recombination signal sequence. We'd have a conserved sequence of 7 amino acids called a heptamer sequence. Sorry, not 7 amino acids, 7 base pairs. This is DNA. Okay, so we'd have 7 base pairs, and then after the V region, we would have a 23 base pair spacer. Okay, and then we would have a mostly conserved 9 amino acid motif, or non-amino acids. I keep doing this. 9 base pairs. Okay, so that's what this RSS flanking the V region would look like. Okay, so this one has a 23 spacer. Okay, and then here, if we looked at these, okay, this would have a nonamer, a 12 base pair spacer, heptamer, 12 base pair spacer, nonamer, and then if you looked at this one, it would be a nonamer, 23 base pair spacer, and a heptamer. Okay, so what you'll notice, or hopefully you may have noticed, is that these spacer regions, they alternate depending on which region you're in. Now there's a rule called the 12-23 rule, okay? And this means that recombination can only occur between gene segments with either with a 12 and 23 spacer. So recombination can only occur between a J and a D, or a V and a D, but these with both the 23 base pair spacers cannot occur. So in this way, right, the cell can't skip over these D regions, okay? And so that's a very important rule called the 12-23 rule. So 12-23 rule. Combination. Spacers. Okay, so that is the gist of what I want to say for how this process is regulated and kind of the rearrangement. And now we're going to get into the step-by-step mechanism in which these regions are actually combined. Okay, so now we're going to talk about the step-by-step process by which the gene segments are recombined. Okay, so step 1 introduces a really important enzyme complex, and that's RAG 1 and 2. We're not really going to talk a whole lot about this particular enzyme, except to say that it plays a really important role because it's the enzyme, and it's actually multiple proteins in this enzyme complex, but it's the protein that recognizes the recombination signal sequence. Okay, so if this is our region in the genome, let's say we'll imagine that this is the heavy chain, so we'll put a D and J, and D and J is the first to combine in the heavy chain. And I actually always remember D and J comes first because DJ is the oldest sibling in Full House. And we have our spacers here, our RSS sequences. I'll color these in, at least for now. okay? And the D region has a 12, right? It's got, let me just pull this back up. It's got the 7 heptamer, so 7 base pairs, plus the 12 base pair spacer, plus the 9, and then this one over here is 9, the 23 base pair spacer, oops, and then the heptamer sequence. Okay. So then what happens is RAG 1 and 2 will recognize these specific sequences. So this is my RAG. These conserved sequences, the nonomer, or so the heptamer. Okay, there's RAG 1 and 2. Okay, and it'll, because of the way the DNA helix turns, it can really accommodate, this spacer really helps it grab onto these 2 regions. Okay, so that's step 1. And step 2, RAG 1, 2, nix the heptamer sequence. So nix the heptamer sequences in the recombination signal sequences. Okay, so what that looks like is now we have this blunt end here. And we have this little nick here. Okay, nick. And this is the 5' region and the 3' region. So it actually puts a little nick right there. Okay, and what happens, right, is, I can draw this out too, we end up with this hydroxyl group, this overhang here. So that's what that nick looks like with the hydroxyl group sticking out there. Okay, so now, so that's step 2. So the next thing that happens is the formation of hairpins. Okay, so this 5' hydroxyl is going to attack, I'll do that in red, this phosphate in the opposite strand, okay? And it's going to make this covalently sealed hairpin. So let me write that all out so you can have it in your notes. So the 5' hydroxyl attacks corresponding phosphate group. To make a covalently bound or covalently sealed hairpin. Okay, so right now we have this hairpin and these blunt ends right here. So 5', 3', 5', 3'. Okay, so then moving on, what happens, step 4, is that there's a ligation event that occurs on these signal ends, okay, and it involves DNA ligase. So ligation, Okay, and so these are ligated. I guess I'll color this. So DNase 4 will ligate these. Okay, and this basically leads to the closure of this piece of DNA, and then this is called the signal joint. And it's basically lost from the cells as an episome. So we've actually excised this piece of DNA. So the DNA for this, if you were to look at the genome, right, it would be now much smaller because we've now lost a piece, okay? Okay, now step 5. Okay, so this is gone. So then step 5 is the hairpin cleavage. Okay, and this is mediated by the protein Artemis, which also relies on other factors, other proteins. Okay, and so it's going to cleave this hairpin right here, okay? And there's 3 possible ways it can cleave. It can cleave and make blunt ends. It can allow for a 5' overhang or a 3' overhang. Okay, so if you're thinking about it, here's what a blunt cut would look like. 3' to 5'. Okay, here's a 5' overhang. And then here's a, 5', 3'. This would be a 3' overhang. So there's 3 ways in which it can have an overhang, okay? Now, something to point out here, so, because we're gonna come back to this in just a second. Let's say the sequences in this hairpin right here, let me get that on the screen, was T, C, G, and A. Okay, and when this hairpin was opened up, let's say we got this overhang, this 5' overhang, we then ended up with T, C, G, A. And so it took the GNA that was at first on the bottom and it popped it up to the top. Okay, so we're going to come back to why this is important in just a second, but it's easy to think about right here. Okay, so the next thing that's going to happen is overhang extension. And let's see here. And this leads to the addition of P or palindromic nucleotides. Or P nucleotides. So due to the nature of the way, let's just take this as an example here, that hairpins are open and they often create these overhangs, you're going to get the addition of a palindromic sequence, which is read the same way forward as backwards. So G, C, A. So here we have T, C, G, A. Oops, that should be a T there. Right? And then we have T, C, G, A read backwards. And so these are the P nucleotides that are added by basically this overhang extension. And this is mediated by DNA repair enzymes. Okay, so I'm just going to point out some things I want you to know, some enzymes I want you to know. We have RAG1 and RAG2. That's important, right? It recognizes the recombination signal sequences and makes that nick in the heptamer sequence, right? And then we have this DNA ligase that gets rid of that signal joint. We have the hairpin cleavage by Artemisius. And then we have the DNA repair enzymes that add the P nucleotides. Okay, in the light chain, this is the end of the light chain. In the light chain, DNA ligase 4 comes back and it basically seals these together, these DNA segments. So in light, DNA ligase 4 joins the segments. All right, so in the heavy chain, and of course, if we were in the light chain, we wouldn't be combining D and J because there's no D in the light chain. But in the heavy chain, a couple different things happen. Okay, so here we go, 8 to 10, and really we're gonna start off here when Artemisius opened the hairpin, okay? So imagine that we have this hairpin T, C, G, A. Okay, and then maybe on this side we have T, A, T, A. Okay, so Artemisius has just opened the hairpin, but in the heavy chain, an exonuclease comes and randomly chews off some of this overhang. So 8 is exonuclease trimming. Okay, so let's say we'll get rid of this one, and this one, and that one. Okay, so then our sequence instead would be just this T overhang, and then we have T, A, T. Okay, so then the next thing is 9. Well, 8 and 9 would be this exonuclease trimming, and then the addition of P nucleotides. So this exonuclease and P nucleotides, so I'll highlight this. So the P nucleotides I'll show in blue. So here we have A and A, T, A. And then for the heavy chain, there's also an enzyme called TDT. So TDT, oops, sorry, TDT. And what this enzyme does is it adds non-templated nucleotides to this coding joint. So it adds non-templated nucleotides, or N nucleotides. So this could be C, T, A, and then G, A, T. Okay, so these sequences were never originally in your genome at all, right? They're just randomly added to this region. And then 10 is that these gene segments are ligated by DNA ligase 4. DNase ligase 4. Okay, so in this one we have a few more important enzymes. We have the exonuclease trimming, right? We have the P nucleotides, which were added by DNA repair enzymes. We have the addition of N nucleotides by TDT, and then the closure by DNase ligase 4. Okay, so that's everything in the heavy chain. So we've talked a little bit about how this process leads to diversity, but I just want to point out some of these once more. So we have a lot of things that add diversity to these lymphocyte receptors. So we have multiple gene segments. For V, D, and J. We have the heavy and light chain pairing. We have the addition of P nucleotides. And then in the heavy chain only, let's change color for that. We have exonuclease trimming. And the addition of N nucleotides by TDT. And so I know this was kind of a long podcast to get through, and we definitely will practice it in class, but just to kind of wrap your head around it, I thought it would be good to go through it before we get into class and before we practice rearranging some of these gene segments. So I look forward to seeing everyone in class, and thanks, bye.